Now that we've done an overview of cellular respiration and the different stages that are involved, we want to go through and talk specific details about each one of these stages. So we'll be starting with glycolysis. Keep in mind that glycolysis is going to begin the breakdown of the glucose, and this is a process that is going to take place in the cytoplasm. The very word glycolysis means splitting a sugar. That is essentially what we're doing here. We will have two stages to glycolysis. We'll have what we call the energy investment phase. In that phase, we will be required to actually invest or spend a little bit of ATP that we already have inside the cell. This is going to help us get over that activation energy barrier. Once we do that, by the time we get to the energy payoff phase, we will begin to get a return on our investment. So at that point, we will begin to make some ATP. We're also going to get some NADH produced when we collect the high energy electrons. This slide right here is very simplified. We're not showing the structures of the molecules, but we are taking a look at the carbons that will be involved in this. So we are starting with a 6-carbon molecule, which is an organic molecule, which is glucose. So notice we have 6 carbons there. We will have a couple different steps take place where we will invest some energy. Now notice in this case we've got 2 ATPs that are going to be broken apart into ADP. If we break ATP, energy would be released. And the reason that we're releasing energy from ATP is because these are going to be endergonic steps. So although the overall process of cellular respiration is going to be exergonic, we do have a few steps at the beginning that are endergonic. We are going to take the phosphates from those 2 ATPs and we will be attaching them on either side of our 6-carbon molecule. And at this point, the name of the molecule will change. It will be called fructose 1,6-bisphosphate. And that right there is going to be a highly unstable molecule. Keep in mind that the more unstable a molecule is, the higher the energy that it actually contains. So this is an unstable molecule. It's very energetic, and the whole purpose of creating that molecule is to assist with the splitting of it. So that molecule will be split into 2 molecules of glyceraldehyde 3-phosphate. Notice that each one of those is a 3-carbon molecule that has a phosphate attached. That will be the end of the energy investment phase, and at that point, starting with our 1 glucose, we have invested 2 ATPs. Notice that we have not made any ATP at this point, so we've just gotten the process started. We will take those 2 glyceraldehyde 3-phosphates that we make, and those will serve as the starting materials for the energy payoff phase. In the energy payoff phase, we will have some redox reactions that take place where the glyceraldehyde 3-phosphate will be oxidized a little bit, meaning we will make some NADH. We will also have a few substrate level phosphorylations take place where we will have kinase enzymes involved, so we will be making some ATP. And our end products of glycolysis are going to be 2 molecules of pyruvate. Those pyruvate molecules are 3-carbon molecules. They are organic molecules, and there is still quite a lot of energy packed inside those molecules. So we will certainly be processing those further to help release that energy from them. If we look at what we made in the energy payoff phase, we do actually make 4 molecules of ATP and 2 molecules of NADH. So if we look at our net or our profit, when we get done with glycolysis, per glucose that we start with, we get 2 ATPs because we made 4, but we used 2. So 4 minus 2 is giving us the 2 ATPs, and then we've also made 2 NADHs. Keep in mind with our energy yields that we want to continually keep track of the NADHs because those will be extremely valuable when we get to the 3rd stage of respiration, that oxidative phosphorylation. That's the overview of glycolysis. We now want to look at the process of glycolysis in more detail and actually show the structures and the enzymes that are going to be involved in this. Keep in mind that this process is taking place in the cytoplasm. Okay, so with the process of glycolysis, we are starting with a glucose. Here we see our glucose structure. It is a 6-carbon molecule. And we will have an enzyme, which is a kinase enzyme, that is going to catalyze this very first step. Anytime we have a kinase enzymes in general basically transfer phosphates. So if we can see that this is a kinase enzyme, we know that it is going to have something to do with phosphate groups or phosphates. And in this case, this one is actually going to be removing a phosphate from ATP and adding it onto our glucose molecule. This step, step number 1, is going to be an endergonic step. It is endergonic because it does require energy, or it's not going to take place. Keep in mind in chapter 8 when we talked about ATP, we said that ATP is useful for running endergonic reactions. That is really the only time we would utilize ATP, is if we had a reaction that was endergonic. Okay, so this happens to be one. The molecule that we produce from this is called glyceraldehyde 6-phosphate because the phosphate is located on the number 6 carbon. That glucose 6-phosphate is a little more unstable than the glucose, so slightly higher in energy because we've taken the energy out of the ATP and we've helped pack it into that molecule. The next reaction is catalyzed by phosphoglucoisomerase. This enzyme is an isomerase enzyme. As an isomerase enzyme, it makes structural isomers. So we will be changing the structure of the glucose 6-phosphate just slightly, and notice that it is going to change from a hexagon into a pentagon. With that little change, it simply changes the name of the molecule. Now, instead of being glucose 6-phosphate, it will be fructose 6-phosphate. The next step is highly similar to step number 1. Here we have yet another kinase enzyme that will be running an endergonic reaction. Because it is endergonic, we are required to use an ATP to make this one happen. And we're going to take the phosphate off of that ATP and we're going to transfer it to our number 1 carbon in our fructose molecule. So then we are left with the molecule called fructose 1,6-bisphosphate because we have a phosphate on the number 1 and the number 6 carbon. That right there is our very unstable molecule that is going to be a lot easier to split. The next step is the splitting step. So in this case, we have an aldolase enzyme. That enzyme is going to break our fructose 1,6-bisphosphate into one molecule of dihydroxyacetone phosphate and one molecule of glyceraldehyde 3-phosphate. Notice that both of these molecules are 3-carbon molecules that each have a phosphate on them. They are slightly different from each other though. In between these two, we do have an isomerase enzyme. That enzyme is capable of converting either of those 2 molecules into the other one. And while initially we get one of each, that isomerase is going to take the dihydroxyacetone phosphate and it's going to convert it into a second glyceraldehyde 3-phosphate. So ultimately, from our glucose that we started with, we're going to get 2 of these glyceraldehyde 3-phosphates, which are what we call G3Ps. That right there is the end of the energy investment phase. Notice that in this energy investment phase, we were required to use 2 ATPs. So that was back here in step number 1 and step number 3. And both of those steps, step number 1 and number 3, those were endergonic reactions. We know that they are endergonic reactions because they require ATP. If they were exergonic, they would not have required energy to actually make them take place. So at this point, we're in a deficit. We have not gotten any energy out of our glucose that we started with. We are going to take these 2 glyceraldehyde 3-phosphates and we will process them further in the energy payoff phase. Okay, so here we have the energy payoff phase. We're going to take our 2 glyceraldehyde 3-phosphates that we have. We do have 2 of them per 1 glucose that we started with. And we're going to begin with a redox reaction. In this redox reaction, the NAD plus is going to oxidize the glyceraldehyde 3-phosphate, and then we do end up getting 2 NADHs. This is a redox step. As a redox step, we should be able to identify the reduction part and the oxidation part. Notice that NAD plus is going to gain electrons, so we would say that it is getting reduced, and the glyceraldehyde 3-phosphate is going to lose electrons, so we would say that it is getting oxidized. 1,3-bisphosphoglycerate is the product of this reaction. This particular molecule has 2 phosphates on it because another phosphate, a second phosphate, will get attached. That phosphate is just picked up from the cytoplasm and attached onto that glyceraldehyde 3-phosphate. In the next step, we have a kinase enzyme. This kinase enzyme will be performing a substrate level phosphorylation. Keep in mind that we said that in glycolysis, ATP will be made by substrate level phosphorylation. So that's exactly what we see happening here. We're going to take a phosphate off of our 1,3-bisphosphoglycerate. It will be transferred onto ADP to yield ATP molecules. It is a kinase that is making that happen. We do have 3-phosphoglycerate produced as a product of that step. The next enzyme is going to be a mutase enzyme. Mutase enzymes are very similar to isomerase enzymes in that they convert between different structural formulas. We have an enolase, which is going to remove some water from our molecule. Then we end up with phosphenylpyruvate. And then in the very last step of this, we have yet another substrate level phosphorylation taking place using a kinase enzyme. And so we get some more ATP produced. This is the end of glycolysis. So notice at the end we're going to get 2 pyruvate molecules. These are both 3-carbon molecules. They're both organic molecules, and there is a lot of energy still packed inside those. This was the energy and the energy payoff phase we saw here. Energy-wise, we made 4 ATPs. Every one of those was made by the substrate level phosphorylation. And we also made 2 NADHs, which are hanging on to valuable high energy electrons from an organic carbon. That's what we made here. If we consider what we made in the previous stage, or what we used, we actually used 2 ATPs. Okay, so we had a negative 2 ATPs from the first stage. So if we put it all together, our net yield at this point is going to be 2 ATPs, and it's going to be 2 NADHs. Okay, this 2 ATPs represents about 5% of the ATPs that we will ultimately make. So we really have not made very many ATPs at this point. Those ATPs are, though, ready to be used inside the cell, so they will be used up probably pretty quickly by some process that needs them. We'll come back to the NADHs a little bit later. Right now they're going to sit in the cytoplasm, and they will be hanging on to those high energy electrons for us. We want to take the pyruvate molecules, and those are going to be processed further. Our second stage of respiration, the citric acid cycle, takes place in the mitochondrial matrix. This means that we must transport the pyruvate into the mitochondria. So as we're seeing on this slide, this would represent our matrix. And keep in mind that the mitochondria has 2 membranes. We have the inner membrane, and we also have the outer membrane. And if we're going to transport the pyruvate into the matrix of the mitochondria, it needs to cross both of those membranes. So we will have a transport protein that is going to assist with that transport. When the pyruvate is moved across the membrane, immediately a carbon dioxide is removed. Okay, so right away we're getting some loss of carbon in the form of inorganic carbon. We will have a redox reaction take place. So notice that we are making more NADH. And then we're going to take the remaining 2 carbons and we're going to attach them to a group which is called coenzyme A. The end result of this is that we get two molecules which we call acetyl-CoAs. That is if we're starting with a glucose. So we're going to have two pyruvates, which means that both of them will lose a carbon dioxide, both of them will lose some electrons when we make NADH, and both of them will get converted into acetyl-CoAs. So per glucose, we've got two acetyl-CoAs that we just made, and we've got an additional energy yield here in the form of two NADHs that we can add to our tally that we just had on the previous slide. Acetyl-CoA is the starting material for the citric acid cycle. So once we do get the pyruvates converted into acetyl-CoA, we will be ready to run the citric acid cycle. Here we see a very simplified picture similar to the first one that we saw of glycolysis. This is a cycle, and as a cycle we would expect it to pretty continuously run. And first off, in this cycle we're going to take the two carbons in acetyl-CoA and we're going to attach that to a molecule which is ready and waiting inside the matrix of the mitochondria and is called oxaloacetate. We're going to take the two carbons and add it to the four carbons, so we're going to get a molecule over here which is 6 carbons. For this 6 carbon molecule, we will have a redox reaction take place. Since this is a redox reaction, we have one thing that's getting reduced, and that will be the NAD+, and we will have one thing that is getting oxidized, and that will be the citrate. Additionally, we're going to have a carbon dioxide removed, and we will be left with a 5 carbon molecule. We have another redox reaction where the NAD+ is getting reduced, and this 5 carbon molecule will get oxidized. Remove another carbon dioxide, and now we shrink down to 4 carbons. Notice at this point in the cycle, we basically put in 2 carbons, and we just lost 2 right here and right here in the form of the 2 carbon dioxides that we released. The second half of this cycle works to convert this 4 carbon molecule back into this 4 carbon molecule. Essentially, we start the cycle with an oxaloacetate ready to go, and we need to finish the cycle with an oxaloacetate that's ready to go, because we are going to be continuously breaking down sugars, and we need to continuously have one ready to attach to any acetyl-CoA that comes into the mitochondrial matrix. Now, when this takes place, we will be making some ATP by substrate level phosphorylation. There will be a kinase enzyme that catalyzes that. We will have several redox reactions over here where we do make some FADH2 and we make some NADH as well. So ultimately, we do get back to our oxaloacetate. That 4 carbon molecule oxaloacetate essentially functions to guide the acetyl-CoAs around the cycle. So we need that molecule always there to be a guide for those 2 carbons. If we look at the yields on this slide, at this point per turn of the cycle, which notice that per turn is 1 acetyl-CoA, we're going to get basically 3 NADHs, 1 FADH2, and 1 ATP. Keep in mind that we will have 2 acetyl-CoAs per glucose that we started with. So if we talk about yields per glucose, we've got to multiply everything by 2. So per glucose, we will have 6 NADHs, and we will have 2 FADH2s, and we will have 2 ATPs. Notice at this point, our carbon will be gone. We did lose 2 carbons on the previous slide when we converted our 2 pyruvates into 2 acetyl-CoAs. 2 carbon dioxides were produced, and now in one turn of the cycle we get 2, and we said per glucose we're going to turn it twice, so we'll get 4. So altogether at this point we will have lost 6 molecules of carbon dioxide. When we finish the citric acid cycle, the carbon will be gone. we look at the citric acid cycle in a little more detail, we're going to take the 2-carbon acetyl-CoA and attach it to the 4-carbon oxaloacetate, and we will get citrate. Citrate is a 6-carbon molecule. There will be some water lost and water gained. We will have a redox reaction where we then produce alpha-ketoglutarate after we remove some carbon dioxide. We have another carbon dioxide removed, and we also have another redox reaction. Then we will have some ATP made. This ATP will be made by substrate level phosphorylation. It will be a kinase enzyme that is involved in that. We will have another redox reaction, and then ultimately one final redox reaction up there. So again, notice that per turn of the citric acid cycle, which would mean one acetyl-CoA, we will make 3 NADHs, 1 FADH2, and 1 ATP. We would have to turn the cycle 2 times to actually get our yields per glucose. If we sum up where we're at this point, in glycolysis we started with the glucose. Keep in mind that that glucose was a 6-carbon molecule. We're going to be taking that glucose, breaking it down ultimately into 2 pyruvates, which are 2 3-carbon molecules. Notice that all of our carbon is still accounted for. We haven't lost any of it. We're going to get 2 ATPs. Those will be by substrate level phosphorylation net, and then we're also going to get 2 NADHs made. We are then going to take our 2 pyruvates, and they will be shipped into the mitochondrial matrix. So this yellow box represents that shipping step. This step is not considered glycolysis or the citric acid cycle. As we ship the pyruvate into the mitochondrial matrix, it will be converted into acetyl-CoA. A carbon dioxide will be removed, and we will also make 1 molecule of NADH. We'll then take that acetyl-CoA. It will be used for the citric acid cycle. And after we go around the citric acid cycle, we will get 1 ATP made by substrate level phosphorylation, 3 NADHs, 1 FADH2, and we'll also get 2 carbon dioxides made. Notice that the blue and the yellow box are within brackets that say ×2. That is all because we had 2 pyruvates out here, and we're looking at yields per glucose because that's what we started this whole process with. So if we sum it all up, our yields per glucose so far is that we have 4 ATPs. These are by substrate level phosphorylation, which would be then kinase enzymes, and they represent about 10% of our total ATP that we're going to make. Keep in mind that we're going to make a maximum of 38 ATPs, so we still really have another 34 ATPs that we need to make. All of our carbon dioxide, though, has been released to the atmosphere. We started with a 6-carbon molecule, and if we completely break that down, by the time we finish the citric acid cycle, we have exhaled 6 carbon dioxides. So the carbon is gone, and all we have left to work with to make that other 34 ATPs are these molecules that we collected previously. So we will use these high energy electrons to make 34 ATPs. Those molecules right there, the 10 NADHs and the 2 FADH2s, those are our starting materials for this 3rd stage. So that is exactly what the oxidative phosphorylation will be using. So if we then move on to talk about that 3rd stage, the oxidative phosphorylation, this is going to take place in the cristae. So we could say in the cristae, you could say the inner membrane of the mitochondria. Now this process is going to have basically 2 stages to it. We're going to have the electron transport part. The electron transport part will be exergonic because we will be releasing energy from those electrons, and then we're going to use it to power the endergonic ATP synthesis. Okay, so this is energy coupling taking place right here. With the electron transport, we're going to go back to the electrons in our NADH and FADH2s. that we collected previously. We will donate those electrons to that assembly line, which is called the electron transport chain, and that will extract the energy from those electrons. As we extract the energy from those electrons, we will capture that energy and use it to power our ATP synthesis by oxidative phosphorylation. First, we'll talk about the electron transport chain, and then we will talk about the ATP synthesis. In the electron transport chain, we are dealing with a whole collection of molecules that are going to participate in these redox reactions. All of these molecules will be embedded in the inner membrane of the mitochondria. These molecules will be at varying levels on this free energy diagram. So notice that as we go down this electron transport chain, or go from one molecule to the next, it is slightly lower on the diagram. As we pass these electrons down this electron transport chain, each passing step represents a redox reaction. If it is a redox reaction, one molecule is getting oxidized and one molecule is getting reduced. So long as we continue to have these molecules, which we call carriers, alternate between a reduced and an oxidized state, as long as that takes place, then we will continue to have this electron transport chain flow, or we will continue to extract energy from our electrons. Notice that when the NADH passes its electrons in, it then turns into NAD+. This means that NAD+ could go back to the citric acid cycle and collect some more electrons. We have essentially a limited supply of NADH and NAD+, and NAD+'s job is to gather electrons, take them to the electron transport chain, pass them in, go back and get some more. FAD and FADH2 are doing the exact same thing. Gather electrons, take them to the electron transport chain. Get some more electrons, take them to the electron transport chain. That is their job. Because this is like an assembly line, all of these molecules must continue to work. So they must continue to pass electrons. If they stop passing electrons, the whole electron transport chain stops. So it is like an assembly line. If one person decides that they're not going to work anymore, nobody else can work either. Oxygen gas is simply the very last member of the electron transport chain. Oxygen gas must be there all of the time to accept the electrons once they've been drained of energy. If it accepts those electrons, then it itself does turn into water when it picks up a couple of hydrogen ions at the same time. If we do not have oxygen gas to breathe, then the electron transport chain shuts down. If the electron transport chain shuts down, then we are unable to extract the energy from our electrons. And if we are unable to extract the energies from our electrons, then we will not be able to make very much ATP from our glucose that we started with. So very important that our electron transport chain continuously runs, and it's very important that we have oxygen gas there to make sure that that happens. Notice that there are lots of redox reactions buried within this. These electrons are going to slowly go from the top to the bottom of this diagram, and that's going to mean that little bits of energy are released along the way, small amounts at a time. The whole function of this electron transport chain is to make it a slow transition from the top of the diagram to the bottom. This is going to mean that energy gets released in manageable amounts, and then the cell is going to be able to better capture that energy to use it for the ATP synthesis process. If we look at the mitochondria, if we draw the mitochondria right here, the mitochondria does have its two membranes. We have an outer and an inner one. And so in my diagram here, this would be my inner membrane, this will be my outer membrane. Inside the very middle, this would be the matrix. The electron transport chain that we're talking about is going to be located on the inner membrane, and there will be a whole bunch of these scattered around, which is one of the reasons why we have that folded inner membrane, or why we have the cristae, because the more surface area we have, the more electron transport chains that we can have. The vast majority of our NADH and FADH2 that we made was right here in the matrix. Keep in mind that that citric acid cycle is taking place in the matrix, and so most of the NADH is being produced in the matrix, and most of the NAD plus is being produced in the matrix from the citric acid cycle. Well, these molecules will make their way over to the electron transport chain, and they will pass their electrons in. When they do, they will then turn back into NAD+, which can go back to the citric acid cycle and collect more electrons. What the electron transport chain will do is pass those electrons from one to the next to the next. Ultimately, at the very end, oxygen gas will grab those electrons, and when it does, it will turn into water. So we have this taking place at all of our electron transport chains. This one, oxygen gas is grabbing the electrons at the very end, and when it does, it turns into water. Same thing happening down here. We've got the NAD plus being generated because the NADH is passing its electrons in. Now, as these electrons are passed down the electron transport chain, we said small amounts of energy are going to be released with every passing step. The electron transport chain is going to try to capture that energy. So if we talk about what it's doing to capture the energy, inside the matrix of the mitochondria, we are going to have a low hydrogen concentration. In the intermembrane space, we are going to have a high hydrogen concentration. So we have a difference in concentration in our hydrogen ions in these two compartments. We talked about concentrations of ions in different locations in chapter 7. We said that molecules want to move, or ions for that matter, want to move high to low. Well, what the electron transport chain is going to do is, as those electrons are releasing energy, it is going to pick up hydrogens from the matrix and move them to the intermembrane space. Okay, so this is with the little bits of energy that are being released. So what we're doing, if we're moving them in that direction, we are moving them from low to high. These ions would not want to move low to high. So this is going to be active transport, and active transport does require energy. So we get the energy from the electrons. Okay, so what we're doing is we are extracting the energy from the electrons. Little bits of energy will be released with every passing step. We take those bits of energy that are released and we use them to pick up a hydrogen from the matrix and transport it into the intermembrane space. What we are doing is we're building up a very high difference in concentration of hydrogen ions in those two compartments. Now, if we look at the ATP synthesis part of all of this, the ATP in the oxidative phosphorylation is going to be synthesized through the process of chemiosmosis. In chemiosmosis, this is an energy coupling mechanism that will use energy stored in the form of a hydrogen ion gradient to drive cellular work. Well, we're going to have a hydrogen ion gradient from the electron transport chain. So the electron transport chain is generating that. And then we're going to be driving cellular work, which will be the making of ATP. Now there is an enzyme, a specific enzyme, that's going to be responsible for this chemiosmosis. This enzyme is called ATP synthase. And ATP synthase has a pretty complicated structure to it. We have a little tunnel in this enzyme. That tunnel is going to allow hydrogen ions to flow through it. So they will be flowing from high to low. So this would be a type of facilitated diffusion. And as they flow high to low, they will cause the wheel on this enzyme to spin. So this is very similar to the way water rolling down a hill would make a water wheel spin. And that spinning action is going to power the enzyme to take ADP and phosphate and put them together to make ATP.